Good afternoon. My name is Prashant Kamath. I'm from Chemistry and Biochemistry and Radiation Lab. And I'd like to welcome you to Notre Dame for this wonderful and exciting topic. And we are going to discuss various aspects of nanomaterials. And Marya Lieberman, who is Associate Professor in Chemistry and Biochemistry Department, will tell us about forces and materials at the nanoscale. Marya has been very active in both scientific outreach and also in nano research. She's a very key part on this bio-nano program in the ND Nano Center. And she has worked on various aspects, and today we're going to hear more from her on the forces and materials at the nanoscale. Marya.
All right, thanks, Prisha. All right. So I thought I would start out by telling you why molecules are like nanomaterials, and then I'll kind of follow up by telling you why nanomaterials are not like molecules at all. And I want to also present an overview of the types of nanomaterials that are coming into use in the consumer arena. And also, because we like stories, I want to tell a couple of stories about nanomaterial toxicity. And so we'll see how that all fits in. I want to start out by taking us down to the nanoscale. And let me start with this beautiful website, which is hosted at the University of Utah. I don't know if Professor Granger had anything to do with this. No? Okay. This is a website that's devoted to teaching children about genetics. But I think it's actually a nice site to just show some of the scale of materials that we're going to be looking at. Because it starts with biological materials or materials of biological relevance where we know what the sizes are. And then you can kind of zoom in. Up here, there's also a little scale. This is a 1 millimeter scale. And so this kind of lets you orient yourself and see how we are doing. So with a grain of salt at 0.5 millimeter, we can now start to zero in on the size of cells. And so large free-living cells and also oocytes, which tend to be quite enormous. We can then zoom down to cells that are part of larger organisms, like a skin cell and a red blood cell, which is on the small end of mammalian cells. And you can see we still haven't arrived at the nanoscale. And so this kind of points out that nanomaterials are going to be interacting with biological materials on the size scale of cells and cell components. So we zoom down a little more, and now you can start to see some cell components, organelles, such as the mitochondrion, which is about the same size as free-living bacteria like E. coli. Now we're getting down towards nanoscopic dimensions. So we're at micron size scales, and of course 1 micrometer is 1,000 nanometers, so we've got to get still smaller. So we'll zoom down a little more. And now we reach the nanoscale. So parts of the bacterium, like the flagella on the bacteria, and virus particles, these are truly now materials at the nanoscale. And of course, if we go down a little further, now we'll be at the size scale of objects like the ribosome, about 30 nanometers in size. These are the size scales of materials that we're making and using in various commercial objects. Okay, so, and of course, if we go down even further, we'll run into individual molecules, protein structures, and then small molecules like a phospholipid, and so forth. Okay, so that's hopefully got us a little oriented in scale. And I now want to just kind of lightly point out some interesting facts about forces that occur in our lives and forces that occur in the lives of particles at the nanometer scale. And so a basic force that we have to worry about is the binding energy of an electron in a box. This is basically the energy of chemical bonds that we're talking about. And that's very strongly size dependent. In fact, here we have 3 different kinds of chemical bonds: a hydrogen bond there, a covalent bond up here, and then in the middle would be an intermediate strength bond, like a coordinative bond. Okay, and these bonds, you can see they're right above the energy that's available from thermal energy at room temperature. And this turns out to be really important for self-assembly and for the lives of molecules, because their lives are lived very close to the edge of thermal noise. So for you and me, walking around in the air, we are not buffeted on a macroscopic level by air molecules and other sources of thermal noise. But a molecule floating in solution, even a fairly big one like a virus particle, is constantly being bombarded by other molecules, and that causes it to move around a lot. That's Brownian motion. Okay? So that thermal bath is always there. And that can actually be useful for helping molecules during DNA synthesis. That's probably helpful in ensuring that the DNA synthesis occurs with high fidelity, that there's a little thermal energy available to help undo erroneous assembled parts. Another interesting force that we experience because we're macroscopic beings is gravitational force. And of course, if one of us were to fall a body length, that would involve a fairly large amount of energy. You would not imagine that outside of a cartoon, somebody could step off a cliff and just kind of hang there in the air. But the gravitational potential energy from falling a body length drops off pretty rapidly at length scales high above a nanometer length scale, to the point where when you get to the nanometer length scale, gravitational energy is not really important. Okay, so the main forces and energies that we have to worry about are electrostatic and chemical bonding energies. Okay, so we have kind of a bizarre world at the nanometer scale. Gravity inconsequential for true nanoparticles. Electrostatics, very, very important in terms of determining whether particles stick to each other, whether they stick to a surface. Diffusion and convection are the main transport mechanisms, and we're in a thermally noisy environment. We're also in a low Reynolds number environment, and that means that the importance of inertia is very minimal for objects even as big as a bacterium. So here we have some E. coli swimming around. Their flagella have been dyed with a fluorescent dye. And let's see if I can get that to run. The movie's not running. Okay, so I'm going to switch to a, this is actually an artificial bacterial flagellum. This is a nanostructure. It's a curved piece of gallium nitride that's fabricated by strain epitaxy, and at the tip there's a little magnet, and a magnetic field is being used to rotate this helix, and that of course enables it to swim through the solution. But if the magnetic field were to be turned off, this particle would stop dead, okay, because it doesn't have any inertial motion. If you were swimming in a swimming pool and you stop swimming, you continue to glide forward. But that doesn't occur with the nanoparticles. Okay, so we're in this kind of strange world. In this world of nanoparticles, the particles move by convection and diffusion, and they can move both in air and in liquids. So nanoparticles form aerosols, which are dispersions of particles in the air. Of course, that presents them as an inhalation hazard. And the particles that are much greater than a few microns can settle out because gravity is still important for them. But by the time you get to 1 micron particles, they can be suspended in air for weeks, causing disruption of airline travel. Ultrafine particles from diesel smoke are of course much smaller and can stay suspended in air for quite a long time. Nano-sized particles also form stable dispersions in solvents, and of course the environmentally relevant solvent is water. And so here the key is their surface charge, because the thing that will break the dispersion and cause precipitation or flocculation are changes in the surface chemistry of those particles. So that's actually can be used to manipulate the stability of the colloid, either to enhance it or to break the stability of the colloid and make particles fall out of solution. Okay. And then I want to, there's kind of the big fleas have little fleas principle that nanometer scale particles can carry smaller particles along with them, and they can absorb molecules on their surfaces and drag those molecules along. And an example that we may be more familiar with is that the small particles of, that are found in smoke are a good carrier for nicotine. And so if you have smoke particles that are adsorbed on surfaces, then that's a sort of a reservoir of nicotine in the, that can be carried along. Okay. So let's get down to nanomaterials and why they're like molecules. So the main reason is that the scale of molecules is pretty similar to the scale of nanomaterials, so they show similar behaviors. And that's just a direct consequence of the types of forces that are acting on both particles. So the loss of the effect of gravity means that we can get these stable colloidal particles in aerosol suspensions. Then the same kinds of forces that can act between molecules to give rise to environmental transport, absorption, and aggregation, that can also occur with nanoparticles and nanometer scale materials. And then lastly, there's a lot of chemical reactions that occur using nanomaterials as the substrate. And of course, one important difference between molecules and nanoparticles is that the reactivity occurs primarily at the surface of the particle, because that's the area that's exposed to the environment. Nanoparticles also, at small sizes, begin to show quantum effects, again, like molecules. Unlike molecules, those quantum effects can be tuned by tuning the size of the particle. So for example, for titanium dioxide that's used in sunscreens, the particle size is correlated very strongly to the absorption spectrum of the particles. Smaller particles, you have a smaller box to put your electrons in, higher energy transitions, and therefore absorption of higher energy light. Okay, so this is one of the factors that's driving the adoption of nano titanium dioxide in sunscreens, is that you can more effectively absorb the rays of light that cause people to get sunburned, and you can leave alone the frequencies of light that cause your sunscreen to make their face look funny, look white or whatever. Okay, when we combine nanomaterials and biological materials, then the level of complexity steps up, because now you have to consider a lot of issues. You have to worry about nanoparticles sticking to your biological system. Perhaps there's specific recognition, which you may actually be engineering into the nanomaterial in order to, for example, target a nanoparticle to a cancer cell. Then there's issues related to cellular uptake. So nanomaterials can be taken inside cells, or they can be rejected and left outside. And obviously, from a toxicological standpoint, they're much less harmful if they never get into the cell. Then there's the standard absorption, distribution, metabolism, and excretion issues that have to be considered in toxicological evaluation of any material. because a living system interacts with the materials that it takes inside itself. Nanomaterials appear to have some potential for oxidative damage, and so that means you need to worry about exposure of the nanomaterial to the biological system and also to sunlight and other sources of energy that can kick off this chain of oxidative damage. And then lastly is immunogenic responses, because most of the studies that are done on nanomaterials to date are not done on whole animals, and often they're done on cells in culture or free-living bacteria that don't have an immune system. And so this is an issue because nanomaterials are at the size scale where if they were talking about a protein, you would be talking about potential immunogenicity. So I guess here's the first little story. This is a little story about C60, which is probably pretty close to being, this is a molecule. You can take C60, make it quite pure. You can purify it to several nines purity. This material has been studied in several kinds of toxicological assays. Of course, it's difficult to study because the solubility in water is pretty low, actually. So people for a while were not very concerned about C60, and then there was a little problem that arose because if you prepare your C60 in a solvent like THF or ethanol, mix it with water, and then pull off the helper solvent, you can get these brown or yellow suspensions. And these are aggregates of C60. So the molecule is quite hydrophobic, winds up sticking together to form these fairly well-defined little particles. And these particles seem to be much more bioactive than individual C60 molecules. So in one study, for example, the inhibitory concentration for bacterium was comparable to vancomycin, which is an antibiotic. Okay, so there's a fairly, in this case at least, there was a reasonable bioactivity. And so there was some concern over this. This actually has brought some interesting features to light. So, for example, when the C60 aggregates are formed, they're formed from a particular solvent. And in this case here, the lower inhibitory concentration corresponded to the aggregates that were made from the THF dispersion. And THF is a organic ether, and it can form organic peroxides. And so in this case, the peroxides appeared to be partitioning into these aggregates. And the peroxides were causing oxidative damage, which in turn damaged the cells. Okay, and so what this shows is that you can have aggregates that look superficially pretty similar, and yet they can have different bioactivity because of, in this case, a relic of their synthesis history. Okay, now that's actually found also in molecular systems. So when the FBI is trying to track a particular source of methamphetamine, often they'll look in the methamphetamine and they'll look for relics of the synthesis, because different synthesis methods produce different trace impurities. And usually people who are making meth are not real concerned about purifying their product. Okay, and so this is just a common feature for molecular species, is that they carry a trace of their history along. And in this case, it seems that trace of their history has an impact on the toxicology of particles. Okay, so now I want to talk about why nanomaterials are not like molecules. So in this case, when you have a simple molecule like a benzene molecule, all benzene molecules are basically identical to all other benzene molecules. But when you have nanoparticles, it's kind of a different case. It's like snowflakes. Every nanoparticle is slightly different from every other nanoparticle. And sometimes those differences are hard to track, hard to keep track of. And so this very high degree of heterogeneity makes nanomaterials more like polymers than like pure molecules. And in particular, the structure and reactivity of the nanoparticle depends on its history. So effects like the residual presence of catalysts in carbon nanotubes and the history of how a sample is prepared are going to affect the dimensions of core shell structures, for example. And of course, when nanoparticles are made, often there's a chemical coating that's applied, and that coating is used to obtain a stable colloidal dispersion. And often the details of that coating have a major impact on the bioavailability of the particle and possibly its toxicological properties. And then the last point is that the structure of nanomaterials is dynamic, particularly when the nanomaterials are interacting with a biological system. So when you put a nanomaterial into a living system, then instantly the proteins start to interact with the surface of the nanomaterial, and there's also reactivity that can be going on. So things can be dissolving, things can be adsorbing on the exterior of the particle. You can sometimes have photochemistry, and so many things can be happening. And of course, to some extent, that is like molecular species. Molecules go into animals and they come out changed. But with nanoparticles, because the structures are more complex, there are more ways for those structures to change. And so I think the heterogeneity of nanomaterials needs to be looked at very seriously. Okay, so this is just a pretty picture showing a lot of different kinds of nanomaterials, and I just wanted to show this just to stress that nanomaterials, even when they share nanometer scale dimensions, there are obvious, there's a great range of possible physical structures as well as chemical structures. And I want to go through some of those structures in a minute. Okay, so when we're dealing with nanoparticles, these are some of the characteristics that ought to be considered. You have a nanoparticle, and then you have to consider a whole host of properties. The solubility of the nanoparticle, okay, which is going to determine whether it dissolves. If you have cadmium selenide nanoparticles, you want to know, is that going to dissolve and release cadmium into the environment? The composition relates to the purity of that nanoparticle preparation and the presence of synthetic adjuvants, residual solvents, stray surfactant molecules, and possibly residual catalysts. So there's many different kinds of impurities that could be present. The charge is determined mostly by the surface chemistry, which is going to have a big impact on the function of the nanoparticle. And so if you have nanoparticles for different purposes, you're going to have different surface chemistries. And so that surface chemistry may make the same nanoparticle present different risks. For example, one may be a flight risk with rapid transport in an environmental context, and another may be relatively stay-at-home because it adsorbs strongly onto soil. And so it's not so much of a problem. Okay. Of course, the smaller you make the particle, the more important its surface becomes because more of the atoms are at the surface. And so the size has obvious impact on properties of the nanoparticle. And then this effective size can be altered by aggregation, agglomeration, and also I would add adsorption on materials that nanoparticle encounters. And then there's some other factors that come in. Okay, so I wanted to go through some industrially important nanoparticles. So a big sector is metal oxide nanoparticles. And I'm going to focus on titanium dioxide, but all other nanomaterials that are made involve ceria, alumina, silica, and iron oxide particles, and I'm sure there's many others, but these are kind of some of the big ones. Bulk titanium dioxide is a major industrial chemical. It's used in paint, plastics, and coatings to give it a white color, and that occurs by virtue of scattering of light. And for these applications, you need a particle size that's fairly large compared to optical wavelengths. So these applications are not going to transfer over to nano titanium dioxide. But there are applications where when you make the particles of titanium dioxide smaller, you get different functionalities or new functionalities. And so we saw earlier that the absorption spectrum of the titanium dioxide shifts when the titanium becomes nanometer sized. And that's used now in applications like invisible sunscreen. So, for example, here this gentleman has half his face with a zinc oxide type sunscreen. But you can now get sunscreens where you put sunscreen on and you don't have a mask on your face. The nano titanium dioxides are used in self-cleaning surfaces like tiles, windows, mirrors, that are where it's important that the surface not have optical distortions. And then there's some applications coming up now in photovoltaics. And so in 2004, the market here was minuscule compared to the bulk market. It's about 40,000 tons is one estimate of the worldwide production of nano titanium dioxide. But I would point out that that is a high-value chemical. It's about 100 times more expensive than the bulk titanium dioxide that's used in paints and pigments. So another form of nanomaterial that's been used now in various products is carbon allotropes. So that includes multi-wall and single-wall nanotubes and possibly some fullerenes, Buckminster fullerene. The main bulk use of carbon, again, is carbon black, which is used as a pigment and also to reduce the resistivity of rubber that's used in automobile tires. And so it improves the wear resistance of the rubber and also prevents you from getting quite as many electrical shocks when you get out of your car, because it helps to leak charge from the car to the ground. Also used in inks. Nanotubes are used in composite plastics, where they serve as a structural reinforcement, but also as electrical grounding. So if you make packaging for sensitive computer chips and you don't want static discharge to fuse transistors on the chip, you can package them in special plastics that are reasonably electrically conductive. And that's a fairly big use now for nanotubes. There's a new use coming up, which is in electronic devices, flat screen televisions, and also flexible electrodes. So carbon nanotubes are trying to take some market share away from indium tin oxide electrodes. And maybe there's an advantage due to the flexibility of the carbon nanotubes. And this is a baseball bat that has some carbon nanotube reinforced plastic in it, and I'm sure you can get golf clubs and bicycles and things like that. So high value sports equipment. 2004, global production on the order of 65 tons. 2007, about 300 tons a year. And just recently, this company called Nano Scale opened a reactor that's producing 400 tons per year of multi-walled carbon nanotubes. A company called C Nano brought a 500 ton per year facility online in China, and Bayer's opening a 200 ton a year facility. So this is a field where more and more of these materials are being made. And once they're made, we're doing the experiment on what their effects are on us. Okay. I want to touch on a couple of materials which are truly nanomaterials and where the uses are not as mature as for the metal oxide particles or the carbon allotropes. And these are the semiconductor quantum dots And I'm also going to talk a little about metal nanoparticles. So cadmium selenide, zinc selenide, cadmium telluride, et cetera. These particles have useful optical properties. In particular, they're excellent fluorophores. And the fluorescence of these particles is very long-lasting and robust and long-lasting. And so the bulk semiconductors are used in photovoltaics and lasers. The quantum dots are used as stable, tunable fluorophores. And they're fairly popular now for use in various imaging applications. So you can buy kits now so that you can attach quantum dots to an antigen or an antibody of your choice and use that to interact with a cell as a stain. And then you can do imaging that way. And this here is showing a mouse that has 3 different colors of quantum dots that have just been implanted under its skin. And this is showing imaging through the skin of the mouse. There's a lot of interest in the nanoparticles that emit in the infrared because flesh is fairly transparent. You can see through a couple of centimeters of flesh. And so there's a lot of interest in these as possible imaging agents for use in detecting tumors, for example. You could imagine targeting the quantum dot with something that recognizes tumors and then and the imaging, and that would be very useful for a surgeon, for example, trying to figure out what they should cut and what they should leave. And so I put these on because although the absolute quantities here are pretty small, I think the use as imaging agents in vivo means that the exposure will be quite large, right? If somebody's getting imaged, then these nanoparticles have to be inside their body. And so it becomes important. Metal nanoparticles, same kind of deal. Of course, gold has a relatively small production. It's used in coinage, jewelry, electronics. The nanoparticles can be quite small. This is a 3 nanometer nanoparticle with an organic coating and some chemical dyes attached to it. These nanoparticles are useful for medical imaging. And there's some other marginal applications like conductive inks. But the pharmaceutical applications here, this is an example here where a mouse, for some reason we always pick on mice, but this mouse has been injected with gold nanoparticles as an X-ray contrast agent because the gold is very electron dense. And so you can see that here the kidneys of the mouse have lit up, and that indicates the gold nanoparticles have accumulated in the kidneys, and indeed over several hours the mouse excretes the nanoparticles. It seems to be unharmed. Here, this mouse has been injected with an iodine image contrast agent, and you can't see very much in terms of where that contrast agent has gone. Okay, so the gold is again one of these items where the absolute amount of gold nanoparticles that's being produced is pretty small, but again it's a high value added product and has potential in vivo applications. I just want to point out that the National Institute of Health is not oblivious to what's going on in this area of nanotoxicology. They have a number of initiatives going on to look at metal oxides, the quantum dots, fullerenes, and the nano silver and gold. And they're doing some studies, exploratory studies going on, and I'm sure we'll hear more about that during the course of the conference. I wanted to touch on a couple of nanomaterials that are coming down the pike. So one of them is graphene, which is an allotrope of carbon. And you can see here is a kind of an infinite graphite sheet. And you can cut out some of the benzene rings and roll them up and make a C60. And you can also cut a different way and roll up and make a nanotube. But if you just cut a little chunk out of the sheet, you wind up with these single layer sheets of graphite. And those have just fabulous mechanical and electronic properties. There's been kind of a graphene gold rush in the last couple of years. This image here is showing not, unfortunately, a single sheet of graphene. This is actually graphene paper, which is made by filtering a suspension of mostly graphene monolayers. And that's, you just filter it on a piece of filter paper, and then you can peel off this composite sheet of graphene. High conductivity, flexible substrate with potential electronics applications. Back when graphene was first reported, the cost was astronomical. And of course, no one was paying $100 million for a square centimeter of graphene. This is just kind of an extrapolation from the tiny, tiny bits of material that were made commercially available. In 2010, you can now, from a company located up in Chicago, you can now buy mostly mono single sheet graphene for about $100 per milligram, available in a stable aqueous dispersion with surfactants on it. So hopefully you can deposit it on your own substrate. And so I'm sure that this price will come down further as the material is used and sold. And of course here the question is, we have these big hunks of graphite. They look a little bit like polycyclic aromatic hydrocarbons, and those have proved to be reasonably potent carcinogens in animals like ourselves that have the enzymes to activate them. And so you wonder, okay, is graphene going to have something of that story? Okay, I wanted to show you another nanomaterial coming down the pike, and this is one you're going to have to indulge me because this is my own area of research. And so I'm working with DNA origami, and these are structures that are synthesized from chunks of DNA. And I'm going to show you a pretty movie in a minute to show you how we make these shapes. But basically, all of these structures are made from the same piece of DNA. They're made from the entire genome of a small bacteriophage. And they're folded up to form different two-dimensional structures using a set of design rules, and also using auxiliary synthetic DNA fragments. Okay, so these are nanomaterials because the characteristic dimensions are certainly on the nanometer scale in terms of length and width, and also the thickness of these particles is only about 2 nanometers. So they're pretty close to being single molecular sheets. Okay, so let me see if I can find the movie. Yep. Here we go. Okay, so this is just showing how the objects are obtained. We take the genome from this virus, and that's just removed, separated from the protein. That's a shot of it on mica, but the assembly is done in solution. We have this long template strand, and to that are added 216 synthetic oligonucleotides. Those are the staple strands. And then the staple strands interact in specific manners with specific base pairs in the backbone of the template strand. So here we see a staple strand coming in and linking together 2 segments of the template strand. And this can link together regions that are quite distant in space, and that enforces a certain fold on the template strand. And so we'll back off, and you can now see more of those 216 staple strands, and you can see the structure of how the template strand is folded up. There's also a little free loop of DNA, and you can see we can also supply some sticky ends of single-stranded DNA, and that enables us to bind these objects together and make larger objects. So, okay, well, that's all very well and good. And then this is a final AFM image showing what these objects look like. This is totally abiological. There's no structure like this in nature. Okay. Okay, so we're interested in this structure as a structural template for molecular electronics. I want to point out there's a recent Nature Nano paper where DNA origami were used to organize carbon nanotubes. So in this case here, there's a carbon nanotube, single-walled carbon nanotube, which is designated here by a blue dot. It's on the top face of a DNA origami. And then on the bottom face, underneath it, there's another carbon nanotube. In this case, it was a semiconducting carbon nanotube. And so through heroic electron beam lithography, the researchers here were able to make electrical contacts to top and bottom carbon nanotubes and demonstrate transistor behavior. Okay, and we're also interested in using these to structure nanomagnets. I think there's a poster or two out in the poster session about nanomagnet QCA. So we're interested in using the origami for that. Now, other applications that people are interested in is just as structural materials. And this is showing some electron microscope images here of designed objects made out of DNA, totally abiological, but using this biomaterial. People have used them as biosensors. So in this case here, the origami have binding sites for where biomaterials, proteins in this case, can adhere, and then the binding can be detected by atomic force microscopy. So you can do single molecule, no label detection. Of course, AFM is a lousy detection method, but another idea people have is to use these as containers. And this is a little box that was designed by a group in Denmark. There's a lid. The lid can be opened or closed. You can put a lock on the lid. And the interior space in here is large enough to hold several ribozymes. So this interior gap here is about 30 nanometers. So it's a significant amount of space inside and possible pharmaceutical applications. So people have started doing cell uptake studies, but there's none of these have been published yet. And so there's some natural questions that come up. Are these going to be allergenic? Are there possible risks from the DNA that's being used to build the structures? And of course, what happens to those nanostructures inside a cell? So these are nanomaterials that I work with. I don't know the answers to these questions. I wanted to go through this little study that's done with heparin. Of course, heparin is a chemical that's used to prevent blood from clotting. And until 2008, the standard potency analysis dealt with the ability of a heparin material to prevent clotting of sheep plasma. Then in 2008, there was a problem. This gentleman here is testifying at a congressional hearing after nearly 100 US deaths due to contaminated heparin materials. And I chose heparin because it's a biopolymer, fairly heterogeneous structure. And in this case, the functional assay of the clotting had been jiggered, basically, by the addition of oversulfated chondroitin sulfate. And this is a material that is able to prevent blood from clotting, but has the side effect of causing severe immune responses and blood pressure changes in humans and also in other large animals. Okay, and so this material had been added to heparin by an unscrupulous manufacturer, presumably in an effort to pass the clotting test. And so that clotting assay was replaced by some more specific assays and also by some tests for composition, NMR and capillary electrophoresis. I want to skip over this and sort of come to a summary. So here the nanomaterials act like molecules because they're similar size to molecules, so there's some similar behaviors, but they're also heterogeneous, and so you can get both variations in the properties, and also there's some really big analytical challenges, which I hope we're going to hear more about during the meeting. And then I also want to stress that nanomaterials are already out there and being used in consumer products, so we have this to deal with. So, okay.
Wonderful overview of the nanomaterial impact.
Just one question.
Yes.
As I hate to admit, the father of that baseball bat.
Yes.
The person who actually made the resin for the golf shafts and everything else. I can tell you that you're absolutely right. The thing about nanotubes, the things about nanomaterials, is they're not going to go into space elevators. They're not going to go into some obscure, exotic application. It's going to be everyday things like baseball bats and golf clubs. And by the way, we turned down the tire application because we didn't want the materials released into the environment. The other thing I'll add is that, in the words of Santayana, a fanatic is a person who, when they've lost sight of their purpose, redoubles their effort. Those plants you see, those 400, 500 tons per year, they're not quite sure what they're going to do with all that stuff yet. As a matter of fact, the German government has a whole program to develop uses for this stuff. The reason why those are being built, I might want to say, is that the reason why Bayer is doing this is Bayer is a German company, and so is a French company. There might be a little rivalry there. But my friends in the industry tell me they don't know where they're going to do with this stuff. So these are some of the things that are out there. I won't say it's always a rational process.
How have the prices of nanotubes been affected by this competition?
That's a very good question, because before it's gone from being obscenely expensive to being only modestly expensive. We've seen price drops in multi-walls, for example, from over hundreds of dollars per gram to now about a few, maybe 10, 20 cents per gram. And that's going to go lower for those people who've had experience in the plastics or the compounding industries. $15 a pound is about the most you can spend on nitrogen on something. So they have to hit that. That's their price point. $15 and you're talking. They're getting it.
I have a question for the questioner and then a question for the speaker. I'm fascinated by that stealth baseball bat. My husband now is working on a memoir on baseball and his father. I know we're right next to Mother's Day. But the serious question is that baseball bat both stronger and more flexible than conventional bats? And will they be allowed by the league? And if they're allowed, will, to level the playing field, as it were, will all batters be required to use a similar bat?
I can tell you a little bit of anecdotal information offline, but to get to the point, the idea there actually was not to create a better bat for hitting. It was to actually reduce the rejects, because conventional bats were breaking about after 700 hits. This can withstand about 900 hits. But to your point about whether you get more action off the barrel, the answer is yes. Matter of fact, you'll notice the company Yeasty Sports does not sell Little League bats or anything like that for that very reason.
So you have an advantage.
Yes.
It's stealth bat.
Question for the question. Are these the same sporting equipment I'm reading now have beryllium in them?
No, this has scandium, and I can't tell you where that comes from, but it's off these shores.
But have you read about the sporting equipment
Yes,
and
yes.
bicycles and a lot of other things that have beryllium as well?
Only three things that scare me as a chemist: beryllium, plutonium, and thallium, in that order.
Yeah, question for the speaker. A beautiful movie, by the way, and presentation. I'm wondering, the little boxes made from the DNA origami for pharmaceutical purposes, could that be used for time release of drugs so that the little lid can become unlocked?
Yeah, the idea with that is that the coding sequences would be put in to respond to, for example, cancer cell markers would be kind of the obvious thing, so that you encapsulate a cytotoxic drug, and then it can associate near the region where it will do some good, and then a signal. a signal from the, hopefully from the cancer cells themselves will make the lid open and release the chemotherapeutic agent.
And then a sort of follow-up question is about the linkages within the DNA origami. How do you put a stop to that? How do you make sure it's not a sorcerer's apprentice type procedure?
Well, the sequence of the template strand is known. That particular bacteriophage is kind of the white lab rat of DNA sequencing. And so everybody with a DNA sequencer starts out by sequencing M13, MP18. And so we know how the staple strands will interact. So you're thinking of kind of like an ICE9 scenario?
This is all predictable. The end is predictable.
The end, this is the structures, the folding of the folds that these structures obtain are predictable. Yes.
I think in light of the keeping up the time, we'll move on, and we'll have more discussion in the panel. We can bring it at that time. And again, my favorite thing is in food products like donuts. All the white stuff you see is titanium dioxide nanoparticles. So